Now we're into sexual cell division. We have everything that we had in asexual cell division. We've got this regular cell. It's got centrosomes, which are also the centrioles. It's just another name. You know, it's got the nucleus. It has all the other organelles. We're just not showing them on here because it would just make it really busy and you wouldn't be able to find what's going on. And then we're going to have 4 chromosomes again. So remembering that we as humans have 46 chromosomes in a normal human complement, well, we're just using 4 because we don't want to try to pack 46 chromosomes into here. If we watch the process of what happens, we're going to find some similarities, but there are going to be some differences because the whole purpose of sexual cell division is to create cells, and it'll be 4 new cells that are going to have genetically different or rearranged chromosomes so that when they are paired with another cell, so if let's say a sperm is produced from this process and it fertilizes an egg from another process, you get a total of 23 chromosomes again. So this happening in a human, we would start with 46 chromosomes at the beginning of sexual cell division and we'd end up with 23. In our case, because we're starting with 4 chromosomes, we're going to end up with half that amount. We'll end up with 2. And I don't want his voice on there. So I'm going to turn his voice off. So we're talking about DNA replication. That process is still the same. This is still interphase. We still have G1, S, and G2. The only difference is that this is occurring in the ovaries or the testes. And though ovary and the testes, as we learned in the reproduction unit, are under the control of hormones, such as FSH. FSH is the one that causes egg and sperm to be developed. Remember, we talked about follicle stimulating hormone. And so the only difference is this is a normal cell. This cell, the follicle cell, that initial spermatogonium cell, have all of the chromosomes from your entire body in it. And those chromosomes are the same as what would be in a skin cell. But the only difference is that this is in the ovary or this is in the testicle, and it is under the influence of hormones. So you still have your DNA replication occurring, and you just saw that happen there. Your identical chromosomes will pair with their copies and join at the centromere. And then G2 happens, and it grows a little bit more again. And then this is where it'll differ, is when prophase occurs. So we'll watch the animation, and you'll see the little spaghetti noodles coming up right away. And those spaghetti noodles are going to condense into chromosomes, the ones that you can actually physically see. And then what happens in prophase is something quite interesting, is that those chromosomes are going to pair up with a chromosome of the same size, shape, and gene arrangement. And so, for example, chromosome 1, there's another chromosome 1, and they both carry the traits such as, I keep always using these ones, but let's say height and hair color. And some of those are tall, some of those are short. It depends on what you got from your parents, because let's say your father's genes were tall, your mother's genes were short, and you ended up with the tall genes, and that's why you appear tall. But you still might have those short genes in your DNA somewhere along the line, but it's just that maybe tall is dominant to short, for example. So what will happen, instead of it going right into metaphase where these chromosomes just all line up like they did back in asexual cell division, these chromosomes are going to switch information. They're going to form a tetrad. They're going to cross over. So let's watch what happens here. So they're forming into tetrads right now, and it's showing it as a pair of homologous chromosomes. And they're going to exchange pieces of information. So a little piece is going to break off of one, and it's going to attach to the other, and the same piece on the other will break off, and it will attach to the opposing chromosome. So we'll watch this happen. Now, it happens completely randomly, and it doesn't happen to every chromosome, and it doesn't happen all the time, and sometimes it happens to more parts of the chromosome than others. And so when you're going through it, you can see right here, it's switching information. Okay? So we keep watching it switch information. Then what happens is, after they've switched information, they pair up at metaphase, just like they did back in metaphase in asexual cell division, but they pair as tetrads in metaphase. And then, when we let metaphase run into anaphase, instead of the actual double-stranded chromosome splitting, the tetrad is going to split. So you'll actually have 2 double-stranded chromosomes going to each side of the cell, whereas before it was the single-stranded chromosomes that were going to each side of the cell. Okay, so we'll watch this, and I've only got about 30 seconds left of this animation. So in part 2, I'm going to continue what's happening in anaphase 1. But just watch how these double-stranded chromosomes are going to the other sides of the cell. You don't have them individually splitting as single-stranded chromosomes. It's double. It's because the tetrad is what separated it, the metaphase plate, rather than the entire chromosome. Okay, so we'll continue in part 2.